Hello everyone, welcome to Merrimack STEM Academy. Today we're going to talk about the central dogma of biology. Central dogma describes how the cell goes from DNA to mRNA to proteins. Understanding central dogma is essential when taking a molecular biology class. So, without further ado, let's get into it. Central dogma starts with DNA. Because each cell only has one copy of its genome, DNA is kept in the nucleus. Transcription is the process of creating mRNA, or messenger RNA, from DNA. mRNA is a temporary copy of a gene, and it leaves the nucleus after being processed. In the cytoplasm, the mRNA is used to create a protein during translation. Now that we have a general understanding of central dogma, we're going to take a closer look at the first process, transcription. DNA is normally wound in a double helix structure. For transcription to occur, the DNA strands must be separated so enzymes can access DNA strands' bases and transcribe the DNA. For transcription to occur, the DNA strands must be separated so enzymes can access the DNA strands' bases and transcribe the DNA. How does this happen? The enzyme helicase separates the DNA strands so transcription can occur. The main enzyme involved in transcription is RNA polymerase. RNA polymerase binds to the promoter, which is a region of DNA. From there, RNA polymerase moves across the DNA strand and uses it to create a complementary mRNA strand. Directionality is key during this process. DNA and RNA strands have a 5' and a 3' end. The ends are named this because each nucleotide has a 5' carbon and a 3' carbon. The mRNA strand must be built in the 5' to 3' direction. This is because the reactive hydroxyl group on the 3' end of the nucleotide can cleave off any additional phosphate groups on the new nucleotide and then form a phosphodiester bond with this nucleotide. So, the mRNA strand must be built 5' to 3'. In this drawing, RNA polymerase is moving to the left, transcribing the DNA. The mRNA must be built in the 5' to 3' direction, so we can label the 5' and 3' ends of the mRNA strand being created. The complementary DNA strand being used to make the mRNA must be anti-parallel to the mRNA. This means that this DNA strand has the opposite directionality. Its 5' end is on the left, and its 3' end is on the right. During transcription, DNA is read in the 3' to 5' direction. This strand of DNA used to make the mRNA has many names, like the template strand, the non-coding strand, and the antisense strand. This DNA strand is called the template strand because it is being used as a template to create mRNA. Let's look at the other strand of DNA. This strand of DNA must be anti-parallel, so its 3' end is on the left and its 5' end is on the right. This DNA strand also has several names: the non-template strand, the coding strand, and the sense strand. It is called the coding strand because it has the same nitrogenous base sequence as the mRNA, except the DNA has the base thymine where the RNA has uracil. At this point, we have transcribed an mRNA strand, but before the mRNA strand can leave the nucleus, it must be processed. RNA processing involves 3 steps. First, a 5' methyl G cap is added. The cap is added by an enzyme before transcription ends. Second, after transcription is finished, a poly-A tail is added to the 3' end of the mRNA. The tail is basically just a lot of adenine nucleotides. Together, the methyl G cap and poly-A tail serve to protect the mRNA from being broken down when it leaves the nucleus. For example, the adenine nucleotides protect the mRNA sequence from being destroyed by enzymes called exonucleases, which can break down the ends of mRNA strands. The third part of RNA processing is splicing. The mRNA strand has regions called exons and regions called introns. Typically, exons are expressed and introns are removed. Once the 3 steps of RNA processing have happened, the pre-mRNA has become a mature mRNA. It is now ready to leave the nucleus and be translated. During translation, mRNA is used to create a protein. Before we talk about the steps of translation, let's talk about the important molecules involved in translation. The enzyme involved in translation is a ribosome. Ribosomes are made of ribosomal RNA and proteins. They have 2 parts or subunits, a small subunit and a large subunit. Of course, mRNA is also important. mRNA is read in sets of 3 nitrogenous bases called codons. Each codon codes for a specific amino acid. Another key player in translation is tRNA, or transfer RNA. tRNA has an anticodon of 3 complementary nitrogenous bases. The tRNA's anticodon allows it to bond to the mRNA's codon. Each tRNA also carries or transports a specific amino acid. The type of amino acid depends on the anticodon. Now that we know which molecules are involved, let's talk about the 3 steps of translation: initiation, elongation, and termination. First, initiation. The ribosome's small subunit in a tRNA molecule will bind to the mRNA strand first. They will basically scan along the mRNA strand, moving from 5' to 3' until they find the start codon. The start codon is always AUG. The tRNA that binds to the start codon always has the amino acid methionine. The first amino acid is a special type of methionine called formyl methionine. At this point, the large ribosomal subunit is added, so the 2 subunits of the ribosome basically sandwich the mRNA. It's also important to note that the ribosome has 3 sites called the E, P, and A sites. This stands for the exit, peptidyl, and acceptor sites. The first tRNA is located in the P site. So, the initiation complex has been formed, and we are now able to move on to step 2, elongation. Step 2 is elongation, as the amino acid sequence grows longer. Remember, the ultimate goal of translation is to use the mRNA to create an amino acid sequence that will be a functioning protein. First, another tRNA is going to enter the A site. Just like in step 1, this tRNA's anticodon and the mRNA's codon will have complementary bases, allowing them to bind together. Here's where the ribosome becomes really important. Each tRNA is carrying an amino acid, and the ribosome catalyzes the formation of a peptide bond between the amino acids. After, the tRNA molecules are going to shift sites as the ribosome moves along the mRNA strand to the next codon. The first tRNA will exit through the E site, leaving behind its amino acid. The second tRNA moves to the P site, where it holds the growing amino acid chain. This is where the process starts to repeat. A new tRNA enters the A site and binds to the next codon. The ribosome catalyzes the formation of a peptide bond between the amino acids. Then, the RNAs move down a site, and the ribosome shifts to read a new codon. This process repeats over and over, growing the chain of amino acids. A protein can have hundreds or even thousands of amino acids. This amino acid sequence has 5 because I'm not willing to draw hundreds of circles. Eventually, the ribosome will reach a stop codon, at which point step 3, termination, begins. The stop codon codes for a release factor. The release factor breaks the complex apart and separates everything. After all this work, from transcription to RNA processing to translation, we have finally created a protein. So, how do we use DNA to create a protein? It starts in the nucleus with transcription, where RNA polymerase creates an mRNA copy of the DNA. The mRNA is processed, adding a 5' methyl G cap and a poly A tail to prevent enzymes from breaking it down. Introns are removed. The mRNA moves to the cytoplasm, where it is translated. tRNA molecules carry amino acids to the mRNA, and the ribosome catalyzes the formation of peptide bonds between the amino acids, creating a protein. Are you now a central dogma expert? Let's put your knowledge to the test. RNA polymerase moves left to right when transcribing the DNA sequence below. Using the DNA sequence, determine the following: the sequence of the other DNA strand, the sequence of the mRNA strand, and the amino acid sequence. Pause the video here. Let's solve this problem. We know that RNA polymerase is moving left to right across the DNA strand. From looking at the DNA strand's 5' and 3' ends, we can tell that the RNA polymerase is reading the DNA 3' to 5'. Since the template strand is read in the 3' to 5' direction, we know that this is the template strand. That makes the other DNA strand the non-template or coding strand. Remember to include directionality here. The DNA strand must be anti-parallel, so this strand is written in the 5' to 3' direction. To get the other DNA strand's code, we just need to use base pairing rules. Adenine pairs with thymine, and cytosine pairs with guanine. Next, mRNA. mRNA is built 5' to 3'. The mRNA is complementary to the template strand, and it has the same bases as the non-template or coding strand. So, we can copy the sequence of the non-template strand, except using uracil instead of thymine because this is RNA. Now, the amino acid sequence. We're going to look from 5' to 3' until we find the start codon, AUG. AUG always codes for methionine. The start codon defines the reading frame, meaning that it determines which groups of 3 amino acids will be codons. Let's look at the next codon, UCU. We can use this codon chart to determine which amino acid UCU codes for. In the left, we select the first base, U. At the top, we pick the second base, C. And on the right, we pick the third base, U. This tells us that UCU codes for the amino acid serine. We then repeat this step for the next few codons. You might be wondering why 3 bases are in a codon. All the proteins in our body are made using only 21 amino acids. So, if we had codons with 1 or 2 base pairs, there wouldn't be enough codons to code for all the proteins. If we had codons with 4 or more base pairs, that would be unnecessary. 3 bases per codon is the perfect number. Because there are more codons than amino acids, sometimes more than one codon will code for the same amino acid. Back to the problem. Eventually, we reach a stop codon, which doesn't code for an amino acid. So, our protein sequence is complete. Thank you for listening. If you have any questions, please leave them in the comments and we will do our best to answer them. Is there a topic you want us to cover next? Let us know in the comments. And be sure to like and subscribe for more videos about biology, chemistry, and everything STEM related.